Catherine.
I think I've unmuted the thing, so hopefully everyone's in can hear, but if they can't, then they shout out. So the first talk this morning is Daniela Egas Santander from the Blue Brain Project at EPFL, and she'll be talking about directionality and reciprocity in brain networks.
Thanks. Okay. So thanks for the invitation to be here. I'm very happy to be in Stockholm again. Can't remember when it was the last time, but every time it's good. Yeah, so I will talk about brain structures and directionality. Okay, so this work would not be possible without the really great group of collaborators. The people on the top are computational neuroscientists from the Blue Brain Project. And the people on the bottom are mathematicians from EPFL and other institutions, some of whom are here. So you can ask them also a lot of questions about this. Yeah, so it's been great to work with these people. And as a caveat, I'm not a neuroscientist, I'm a mathematician. I belong to the second row. And because it's a very diverse audience, I thought I will, instead of doing lots of details about some specific project, give you some idea of how we are thinking about these things in like a broad perspective rather than a detailed perspective. And in particular, how directionality plays a role in all the things that we're doing, whether we take it into account or not. Okay, so probably all of you know, there's this very strong hypothesis in neuroscience that the way the brain is connected, its connectome, its brain architecture, drives its function from very low cognitive functions to very high cognitive functions. So natural questions are, what are the wiring rules of the brain? And how is this structure shaping the function? And through my exploration of neuroscience in the last couple of years, I realized that people try to answer this in two different ways, either in a very data-driven way, where everything is as approximately as possible as the physiology tells us it is, or in a very concept-driven way, and like, okay, this is my hypothesis of what the brain is doing conceptually, and I'll build a model that does that and try to bridge these two things together. I think they lie on opposite sides of the spectrum, but they're complementary to each other, and one can play going back and forth between these different things. And so these things divide these questions in some axes. And yesterday we saw a lot of things in the top right bottom. So there were these oscillators, which were conceptual models of brain work, how brain works. But you can also think of Hopfield networks. You can think of threshold linear networks or any of the architectures built in machine learning. These are all concept-driven things that harness the structure to build in some function. So I'm not going to talk about that today. Instead, I'm going to talk about these three other corners and tell you how they interact with each other in some specific way. And I want to start with the top left, this data-driven perspective of the wiring rules of the brain. And for that, we are very lucky to be alive right now, because incredible advances in engineering and science have made a plethora of connectomes really available to us in different scales and different animals. So from micro scale, where we saw some yesterday with epilepsy, macro scale for human, for macaque, for mouse, but we also have fully traced connectomes in the micro scale. So how neurons are connected to each other for different species. We have a full one for C. elegans, a mostly full one for Drosophila and Drosophila larva. I should stop calling it baby, has just come out. So we also have that. And in the place I work with in the Blue Brain, what we do is digital reconstructions of such connectomes. And we have these for different parts of the brain, for different species, but mostly for the rat. Right, so we have this huge playground to explore from this biophysically detailed and approximate point of view, what are the wiring rules of the brain? And I will tell you a little bit on how we are doing this at the Blue Brain, but the tools that I will tell you about apply to anything really on this row, and modifications of that should apply to macro scale. These are really connectomes of different nature. They're generally full matrices that are weighted, while these are sparse matrices that sometimes are weighted, sometimes are not. So the tools are not the same, but they are related tools that can be used. So as part of that, at the second half of last year and the beginning of this year, we finally released a data-driven, biologically detailed model of the full SSCX except the barrel of the rat somatosensory cortex, so the part of the brain that takes input stimuli and sends it to the rest of the brain. And because I'm not going to talk just about this, but rather about the methods, I'm not going to spend a lot of time explaining this model, but rather just tell you some hints of how big these things are and what do we know about it. So just to give you an idea, the model has about 4 million neurons and about 4.5 billion connections. Some of these are local connections, so neurons that are connecting to each other because they're in close proximity, and some of them are mid-range connections, so like long connections that are made across this volume. And we have simulations of in vivo-like activity and replications of experiments done in this volume. That is as much as I'm going to say about the model itself. But now you have such a model, or you can think of C. elegans or Drosophila or any other kind of connectome that you want. And you want to say, I want to quantify its structure. We heard a lot yesterday, we know brain networks are not random. But how do we know that? And how do we quantify how not random they are? So let me just tell you something that we already knew from small volumes that we now know for large volumes. And I think most of the people here are not coming from a topology background, so I'm just gonna remind you or tell you some of the motifs we're looking at if you're a topologist. So one way of thinking whether a graph is random or not is to look at motifs. And if you're a topologist, you very much like to look at simplicial motifs. So that's one reason. Another reason to think about it is because simplicial motifs are kind of small embedded networks that are all-to-all connected and all feed forward. So they're very special for the transfer of information. So what is a simplex? A simplex is a motif that is all-to-all connected, but in a directed way. So there's always one source neuron that maps to all others. Then there's a second neuron that maps to all the rest, a third neuron that maps to all the rest, etc. And the number of neurons minus 1 is the dimension of the simplex, because we can encode it like that if we would build a space out of this, but there will be no spaces today, unfortunately. Maybe later, I think later there are some spaces. So for example, this here, a single edge in your graph is a 1 simplex, a triple of nodes all-to-all connected in this feed-forward section, and this feed-forward fashion is a 2 simplex. This is a special kind of a triad, if you've seen triad analyses in neuroscience. This guy here is a triad, which is not a simplex, because it is not feed-forward. There is a source, but there is, I turn it around. It is a simplex. Ah, no. Okay. Ignore this one. If I will flip this arrow in this direction, it would not be a simplex. Mistake. So this is a simplex. And this guy is a 3 simplex. There's no blackboard. We cannot fix mistakes.
Around the blackboard, the back of it.
Yeah, it's like super difficult. So one reason we care about simplices is because they're like a natural object in topology. Another one is because they're feed-forward networks. But the very important one is because random graphs with a certain structure, for example, say Erdős-Renyi on X number of nodes of density blah, will have very narrow distributions of how many simplices they will have in each dimension. We know this analytically for Erdős-Renyi graphs, and we know this through experiments for other types of graphs. So it's a way for you to tell, is your graph of this random type or is it not? Because it will lie way outside of the expected distribution. So we have counted simplices for this ginormous graph. Big hats off to Jason that makes our code more and more efficient, that allows us to do this. And we get something like this. So for example, here, this curve is the number of simplices in this ginormous graph. We have simplices up to dimension 7. These are just local connections. And everything here are many samples of random graphs of a different type. This is the average plus a width of the standard distribution. So the simplest one is Erdős-Renyi of the same density. Then we have stochastic block models. Our model is very biophysically detailed, so not all neurons are the same. They have shapes, which are called morphological types, and they have different probability of connection with each other. So we also do a model where we do that. We say we have these blocks of connectivity between the different M types, but then besides that, they're randomly connected. We know that the graph is not like that. We also say configuration model, which is a type of graph where the degree sequences are fixed. So if we sample graphs at random with the same number of edges in the same degree sequence, is our graph of the same type? And the answer is no. We're very, very far away from the expected values for such a graph. The closest thing that we have to our model is what we're calling distance-dependent block. So it's a random graph where the probability of connection decays exponentially as you move far away. And the parameters for the decay depends on the morphology of the neurons. But even such a detailed graph is not the kind of graph we observed here. And I have to say, this analysis have been done before on a very small column, which is 1/100th of what we've done here. And it has been, the presence of such motifs and local connectivity has been verified in the lab through wet lab experiments by doing patch clamping. So even though you might think this is just a digital reconstruction, such high dimensional motifs actually do exist in the brain. And if anything, we're underestimating them. Yeah?
I was missing for the big graph on the top right, is that for the connections, was that made on EM experiments, or is it one of those models from the blue brain where you have a statistical model to link them?
Yeah.
Which of it is?
Is the second.
Okay.
Is the second.
So it is not data, it's a model.
It's a model. Yes, it is a model. You can do the same analysis on C. elegans and you'll find different results. And this is data. And this is what I was trying to say before. This like presence of high simplices has been found in the data through patch clamping. But it's really hard to measure the actual connectivity of 4 million neurons. So this is just a model. And now the nice thing about this new model is it allows us also to look at the graph of mid-range connections and see, is this object different or similar to the one from local connectivity? And mid-range connections is here in orange. And it is also different than any of the random controls that we find. In this case, it's closest to the configuration model, but it's different from local connectivity. And also adding them together, like adding simplex counts together, is not the same as looking at the full graph. There's really a lot of structure in their interaction between them. So this is just to give you an idea of how you can quantify if your network is random or not. And we've done a lot of analysis of what this means in terms of biology, but because I wanted to connect to these three areas, I'm going to be very brief on this, and I'm just going to tell you. Yeah.
What was the difference in the simplex counts you did? What was the local simplex and long-range simplex?
Yeah, so this model has two types of connections.
Oh.
Some connections are just close, like the way the model is built. Some of them are just, is my tree close to the next one and can actually make the connectivity, and the other ones are mid-range connectivity, so they're tracing long across the volume. Sure. Yeah.
Now that I know it's a model, I have a question, but maybe you explain the model later.
No.
No, okay. Then could you say briefly what are the features of this model that you have on top, say, of a trivial Erdős-Renyi graph? Yeah, so I didn't want to spend a lot of time explaining the model because then the talk would be just about that, and I wanted to do it something else. But let me tell you very succinctly the idea. The idea is a very data-driven model to try to approximate the physiology. So they have data about how many, so the cortex is divided by layers, and there are different morphologies of neurons that live in every layer. They know this from measurements, and they have different densities in each of the layers. So they use this to populate an actual geometric volume with these neurons. Then there's a separate pipeline that actually generates the shapes of these neurons. So they're not thinking of them as point neurons, but actually as trees embedded in 3D space, which are now embedded in this volume. And then there's an algorithm that tells you, okay, if I'm close enough, do I connect? Do I not connect? Do I prune? Do I not prune? And then there's a separate one for the mid-range, which is there's a certain folding algorithm that is known to model these long-range connections, and then they're modeling the long-range connections from that. So it's not, I mean, it is at heart a generative process, but it's not a basic one. It's one that has taken several years to implement in a single model. Yeah, I got it. Yeah, thanks. Yeah.
Okay.
Is it computationally possible to put all the local and long-range, mid-range connections into one huge network and compute the simplex counts?
Yeah, the combined we have done, and the combined are here in gray.
Okay.
And I put them here. Yeah, I didn't explain that. Thanks. So this is putting both of them together. Orange is the mid-range, and the blue is the sum. So that's what I was saying. The sum is very far away from what happens in the combined. So there's actually a lot of things coming from the fact that these two networks are living together in the same set of nodes, which you can then try to explore what does it mean in terms of the activity of this network. And I'll just give you a hint and leave it there. So one thing you can do is this notion of whether a neuron is or is not in a simplex of a certain dimension, and on how many simplices of a certain dimension is actually giving a notion of centrality of this node in the network. So you can now, instead of thinking just as your network, you can think as your network with different weights on the vertices, which are like the node participation. And the basic one is you can just say, what is the maximum dimension simplex a neuron is in? And this is this plot here. So this is a flat projection of the volume I've shown you before. And the color tells you what is the maximum dimension the neurons in this region belong to. And so this is local connectivity in green, and this is mid-range connectivity in orange. And so you see local, you have almost maximal dimension everywhere. There are very few neurons that belong to simplices of dimension 7, but they're scattered kind of across the volume, and then everything else is pretty even. While mid-range connectivity has some hotspots of central nodes, and then some pretty empty nodes across the volume. And then, so this is a way to explain what is the difference between the networks that is not just simplex counts, like this one is an heterogeneous network that is more or less everywhere the same, and homogeneous, sorry. And this one is a heterogeneous with pockets of connectivity and pockets of sparsity, pockets of connectivity and pockets of sparsity, and they're interacting together. And you can try to play around with what does this mean in terms of the activity if you want. Yeah.
Can you exclude that this is not just where the folding gets closer, for example?
Not always, but sometimes, yeah. But definitely some of the hotspots here these are related to these foldings. But not always, yeah.
Okay, thanks.
Okay, that
yeah.
probably can be checked experimentally, no? But there will be like places or not. It may be difficult to check, but there are these.
Yeah, I don't know what much can be measured really experimentally on this level. So I think, so one of the.
Like a check that there's like no mid-range.
Yeah.
Connections coming, neurons from the, in some patches, right? Like, well, there's no.
Yeah, we would have to ask a wet lab experimentalist if this is actually possible, but I think this is one of the reasons looking at these models is interesting. It will ask questions that you can then test in the lab. So a couple of years ago, when these models found these highly connected motifs of dimension 7, then people were thinking that those do not exist in the brain, but it motivated the experiment to actually go and see if they actually can find them, and they found a lot of them. So maybe something like this heterogeneity could be done, but I don't know the feasibility of such a thing. Like, we will have to talk to an experimentalist.
I have a question here.
Somebody, yes?
I was just wondering, like, what's the distribution of degrees if you just render the degree of each node?
Yeah. So both are long-tail distributions, but this one here is much more radical.
But does it match with these hotspots as well? Like, I'm like repeating the previous question, not folding, but the degree, whether the degree explains this.
So it matches for some of them here, but not for all of them. So some of them here have this really crazy out degree. So we are calling them monster neurons. And this explains a lot of why there are these high dimensional things here, but not everywhere. And that's why, whoops, something happened. Okay, that's why here, the closest model to mid-range is actually configuration model. So it's a random model that has the same degree sequences, or approximately the same degree sequences, but it is not explaining, I don't know how to quantify it, but it's not explaining at all the whole structure of the network, because the mid-range connectivity goes up to dimension 15. But the configuration model will only go up to dimension 6. So it's not all, but it's definitely part of it. It's definitely a very important part of it, as opposed to local connectivity, which is also long-tailed, but not so radical. But these long tail distributions of degrees are found also in experimental data. Like this is expected to have these outliers of connectivity. Does that answer the question?
Yes, it does. Thank you.
Okay. Yeah, and this is just to give you a hint that these things are actually related to the activity. Before, it has been found that neurons that belong to simplices have higher correlated activity, and this increases with dimension. And now what we can also show is these neurons are of different types. So if we split them by population, excitatory, inhibitory in this layer or in this other layer, and we look at their average activity versus their average node participation, so how many simplices this node is part of, there is a strong correlation between activity and this like centrality metric. And this is for simplices in dimension 1, which would be degree, which was the question before. And this is simplices in dimension 6, which you can think of as a higher version of degree. And the interesting thing is the correlations are always high, but they generically increase with dimension. So this higher or generalized version of degree becomes more meaningful for function as dimension increases. So I hope I have justified that we have tools to determine if a network is random or not of a certain type, because if you just say the network is not random, you have to add some meaning to this sentence. So it's not random of this type, it is not random of this other type. We have tools to say that, and we have tools of using these metrics and relating them to function in a certain way. Okay. But the simplices are not the only kind of directed motif that exists. Another one that occurred a lot yesterday. What time do I have to end? What?
Quarter to.
No, sorry.
Okay, so another one that occurs a lot, and it appeared yesterday, and all these random matrices as overrepresented in some way, are reciprocity of connections, reciprocal connections. So it's a natural question to ask, how are these reciprocal connections related to simplices? And a simple thing you can do, and we have done for a smaller volume, not for the whole 4 million one, because that is a very something's happening. Okay, I don't know. I think we have done for the smaller volume of only 32,000 neurons or so, is you can take the graph and now look at the subgraph on all the edges that are in at least a 2 simplex. That gives you a smaller subgraph. Then you can say, I'm going to look now at all the edges that are at least in a 3 simplex. It's a smaller subgraph. At least in a 4, at 5th, etc. So you get a nested sequence of subgraphs given by the edges being in these higher dimensional motifs. And at each dimension, you can ask, what is the percentage of reciprocal connections? And this is this curve here. And so you see that this percentage of reciprocal connections increases with dimension. This other curve here is if I don't look at all simplices, but only maximal simplices, so simplices that are not contained in a simplex of higher dimension. So this leads you to believe that reciprocal connections preferentially lives in simplices, and this preference increases with dimension. But for reasons that I hope will become clear later, you might believe that actually this is a combinatorial trick and it's not the real thing. So you want to make some sort of control. And we'll do this control because it's a cheaper control, which is for simplices in each dimension, what is the average number of reciprocal connections? So in blue, this is what we find in our network. So in dimension 6, the average number of reciprocal connections is 1.75. Okay, and it increases with dimension. The other ones are random controls. So the most basic one, Erdős-Renyi, because everyone always has to do that, you make an Erdős-Renyi one, and then you add reciprocal connections because this guy will have less than your model. You add them at random, and you do the same count. And then we do better controls or closer controls. So the green one is you take your original graph, every reciprocal connection at random, you choose a direction, and delete the other one. So you end up with a graph with no reciprocal connections. And now you introduce them back at random anywhere in the graph. So you end up with a graph with basically the same underlying undirected structure, but the reciprocal connections are kind of everywhere. And then you do the same counts, and this is this green line here. And here I just divided the blue by these ones just to show you that indeed reciprocal connections prefer to live in simplices, and this preference increases with dimension. And now you can say this is just a model. Maybe it's a feature / bug of the model. But we have done this in C. elegans, and in C. elegans, this preference is even larger. We cannot do it for the real adult Drosophila because that connectome has too many simplices, but we should probably try on the larva to see if we can actually list them and see if it's also present there. Right, so these two kind of canonical directed motifs and brain networks, they are not only present in an overrepresented way, the way in which they're present are related with each other. I hope I made that point clear. Is everybody good? I'll take the silence as a yes. Okay, so this brings a lot of questions from a concept-driven point of view of what are these wiring rules of the brain, which you would try to answer with either maths or physics or whichever tools it is that you are used to. And I'll try to just explain some of them. So a very basic one is we say, okay, these random graphs are not Erdős-Rényi, they're not the stochastic growth model, they're not distance dependent, they're not configuration model. Maybe they're just random graphs with fixed simplex counts. So if I could take the set of all random graphs with fixed simplex counts and I will sample them at random, these brain networks will be one of those. So you need a method to either generate such graphs or sample them uniformly at random. This turns out to be quite a difficult problem. First tackling of this problem was done by Unger and Krebs, and I actually checked, Florian talked here last year, and he talked exactly about that. So they do a first approximation of this. They sample graphs uniformly at random with fixed simplex counts, with the caveat that they fix the underlying undirected structure. So they cannot do it in general, they do it in this very specific case. But already with this, they can say that the brain networks are not of this type. Yeah, but this remains an open question. If you're a topologist, you say, okay, forget graphs. Can I just sample simplicial complexes, which is our generalization of graphs? In graphs, you say these two nodes are connected to each other. In simplicial complexes, you say multiple nodes are connected to each other. So, or hypergraphs, if you've seen them before. Can I sample simplicial complexes or hypergraphs with fixed simplex counts uniformly at random? We're trying to figure out ways how to do this. It's not so simple, but maybe we can get some approximations of these and do like rich club kind of analyses with fixed simplex counts. What I've mentioned before is this is a tool to tell you if your graph is random or not, because there are very narrow expected values of motifs for a specific kind of random graphs. We know this analytically only for Erdős-Rényi, actually. So it is also an interesting question in stochastic topology. Can we do this analytically for other kinds of models? I'm just listing some interesting questions. And then one that I want to tell you about because it's super cute. It's motivated from neuroscience, but it goes somehow deeply into math, is how do adding reciprocal connections affect simplex counts? And that's why I was telling you before, you might be confused. Am I seeing something real or is this a combinatorial trick? And also in the work of Unger and Krebs, they find that the hardest part of the problem, because they start with a fixed undirected graph, and then they just have to assign directions and assign reciprocal connections and keep the simplex count fixed. A big part of the problem is where to put these reciprocal connections. And I will try to explain why this is complicated, but interesting. So if we want to make it super simple, so we forget we're doing neuroscience for 6 minutes and we're just playing a game. And we're saying, okay, forget 4 million neuron graphs or whatever. Let's start with something very simple. I will start with a simplex. So a set of n neurons that are all-to-all connected in this feed-forward fashion. That's it. Start with something of size 2. And now I add reverse edges. Well, how many n simplices are there in this end graph? This is the question. Does everybody understand the question? Okay, who understands the question? Thank you. Okay, good. Good. So, for example, in dimension 2, this will be a 2 simplex. And for just visualization purposes, we can think of these as the matrix, right? I can order. So if you haven't seen a simplex before, an interesting, useful fact is that every simplex has a longest path, which goes from the source to the next source, if you delete that to the next one, and to the target. So, and this longest path is unique, and it has length, the dimension of the simplex. So in these two simplex, this is the longest path. So if I order my nodes 1, 2, 3, and then I do the matrix 1, 2, 3 for this graph, what I will have is set of ones, which I mark in yellow above the diagonal, and zeros below. So adding so how many simplices are there here? 2 simplices, there are 1. I really hope I didn't mess up the arrows last night on this one. So here, I add up this reverse edge. And I ask how many simplices, 2 simplices are there in this graph? Anybody knows? Well, I'll tell you, and then you can think about it. There's still 1. There's only this one that I've created because this new edge has generated nothing. Okay, but what if I put it here instead, which is what I did the mistake in the beginning of the talk. I put it here. Now, suddenly I have two simplices. I have this one, and I have this one. I have two simplices. So if I want to answer this very simple question, I start with a simplex and I add reverse edges. How many simplices are there? It doesn't only matter how many I put, but where I put them. And when I start putting more than 1, they start interacting with each other and it starts becoming a bit crazy. So just to tell you, for you to have some fun. So now you start with some simplex of size n. How many n simplices are there? There's 1. Another basic counter, which is just so, if we add a reverse edge on consecutive nodes, so from I plus 1 to I, we will have 2 because we can flip this in the longest path. That's what I just showed you in dimension 2. And the other basic count is if you add all of them. If you add all of them, suddenly you have n factorial because you have all possible ordering of the vertices will give you a path and it will give you a simplex in this graph. And then you can say, okay, what kind of stuff happens? So I'll just show you 2 counts that I think are very fun. So you start with a matrix of size n, and then you start adding reverse edges along the lower diagonal, and you add n of them. Then what you have is Fibonacci numbers of simplices. So if you add n edges, you have fn plus 2. This should not be obvious to you at all. You just have to believe me. I think it's fun to see what can happen. Another fun thing that can happen is if you have a matrix of size 2n, and you then put this block of size n and n of this shape of reverse edges, what you will have is Catalan number of 2n simplices in this graph. This again should not be obvious, but it's just like all these crazy numbers are coming. So how is this happening? Because I started with a very simple, naive question coming from neuroscience. I add reverse edges, what happens to the simplex counts? All these crazy numbers are happening. Why is this happening? Well, it turns out that this is related to a deep mathematical question that people have been looking at for a while. So if you haven't thought about this before, just let me tell you about this. No, let's go back so you don't read it. Okay, so if you have a simplex, as I told you, you have the longest path, so you can think of this as giving you an ordering of your vertices. So when I start adding reverse edges, I'm kind of saying, I don't care about the ordering of these 2. I don't care about the ordering of these 2. So instead of having a total order of your vertices, you just have a partial one. And when you ask how many simplices are there, you're asking how many ways can I order all these vertices respecting the restraints that are there. And that's why this is equivalent to, if you haven't seen this, it doesn't matter. But if you have seen this is the same as counting the number of linear extensions of a poset, and this is the same as counting the number of inversions of a certain permutation. So if you haven't seen this, forget about it. You can instead think about what applications there are there. There are applications in sorting and algorithm planning, because I just told you this is telling you like a partial order, and you want to say, okay, I have these things that have to happen in this order, and the rest can happen in whichever way. I'm basically counting how many simplices there are in this graph of tasks. It has applications on social sciences. When you have a partial order, you have to rank people and you have to look at a total order of them. So you can look at all possible orderings and then kind of average. It has applications to computing volumes of complex polytopes coming from posets. If you are in math, if not, ignore that last application. So because it's been studied for a long time, we also know quite a lot about them. And the first thing that we know is, unfortunately, finding this out is P-complete. So we're not going to solve it very easily for large 4 million neuron brain networks. There are some approximations that can be done in polynomial time. There are some upper and lower bounds, and there are some exact formulas for certain cases. And if you know posits, there are cases given by the structure of the Hasse diagram. So our little contribution here is giving a set of formulas for some specific cases, and I'll just try to tell you the idea of what cases there are, and yeah, and then take you to the last part. So the idea is to divide and conquer. So I already told you, if I add this guy here, I'll have two simplices. But because I can flip these two vertices. But if I add this reverse edge also here, but separate, what I have is 2 times 2 simplices, because I can flip nobody, I can flip these two, I can flip these two, or I can flip both. And that idea is generic. So if I have 2 disjoint sets, one green in U and one purple and red that are separate from each other, then the number of counts of simplices is how many there would be if I add U times how many there would be if I add V. So the idea is divide and conquer. So instead of thinking about exactly where these reverse edges are, I can think of the structure of this graph of reverse edges and seeing if it's separate in separate ways, and I can just look at the separate parts. And this idea is encoded in what we call modular partition. Everybody calls a modular partition, which everyone has probably seen because they are very common in neuroscience. So basically, a modular partition of a graph is a subdivision of the nodes into sets. So here we have the red, the green, the blue, the pink, such that the neighbors of all the reds are the same, the neighbors of all the greens are the same, etc. So all the reds connect to all the greens, all the reds connect to all the blues, none of the reds connect to the pinks. So then I can summarize my graph by its skeleton. So I collapse all the subsets, which are called modules, into a single vertex, and modules that are connected to each other, I connect in the skeleton. Of modules that are not, I do not. Okay, so very roughly, I'm slightly lying, but lying is allowed in a talk. We basically can give formulas for simplices on the cases where the graph of reverse edges can be given as a modular partition of these 3 shapes. So either the modules are connected to each other in a line, or the modules are connected to each other in a binary rooted tree, or the modules are connected to each other in this sort of necklace. And just to interest you, because of how much interesting and complicated math can come up from a naive question in neuroscience, I'll just show you for 2 seconds what these formulas look like, so you have an idea that this is not a trivial question, but you don't need to remember them at all. But then if we want to go back to brain networks, we have to say, okay, we looked at this very simple case. Can we use this approximation somehow to extend this to a general base graph, not to a simple simplex on n nodes? Maybe not. Another question is, how can we find efficient modular partitions? This is not simple. If people are studying modularity in networks here, they will know this is not so easy. And the big question is, we actually added reverse edges in some fixed shape that we can understand. But the question in neuroscience is, if I add them at random, what would be the counts? Can I have expected values to the counts? So this will give me analytical answers of whether the brain has reciprocity placed at random in the network, or if it's really determined by these simplices. We know experimentally that it is, but we don't know it analytically. So let me use my last 6 minutes to give you an idea of how playing around with these concept and data-driven understanding of wiring rules of the brain can give you some useful tools to study the relation of structure and function. Okay? Right. So brain networks are not random. We have quantified them, or maybe they are random of some kind that we don't know. They're very specific. Why are they like this? And an idea is that there's a delicate balance between a network being efficient and a network being robust. So if you are efficient, you encode a lot of information with few neurons. The activity of the neurons is all uncorrelated. If you are robust, most of your activity is correlated, but if I delete a neuron or if I delete a connection, not much happens, right? So if we would take this to the extreme of our intuition, the most efficient thing will be having independent neurons, each one coding for different things. But then if you delete a neuron, you're screwed, you forgot the name of your mother. But if you are robust, then everything is connected equally to everything. And if you delete something, then it doesn't matter. But this is very inefficient, not only because the activity is all highly correlated, because also we know that wiring in the brain is expensive. We cannot have such dense networks inside the brain. There simply isn't enough space. So we need to find sparsity also for biological efficiency. So there is a balance between these two. And our intuition is that networks in brains are like subsets of very dense things and then sparsely connected to other subsets of very dense things, which is what we've seen like this with this modular hierarchical structure in brains. Now, what I will try to convince you is that we can use these topological ideas to make this precise for certain networks. So we have very few connectomes, even though we have many, like for what we knew had 15 years ago, but we don't have 100 human connections, 100 Drosophila connections. We have very few. So we cannot experiment on a connectome level. We have to experiment on subsets of these. And on a way we can do this is we can, instead of looking at the whole connectome at the time, let's look at neighborhoods at the vertex. So let's look at all the vertices that connect to a single node and how they're connected to each other. And neighborhoods will look very different. Some of them, like this blue one, will be very sparse. And some of them, like this purple one, will be very dense. So they will give us an idea of different varieties of networks that exist in our connectome, and we can use this to evaluate our metrics. And I hope I have used almost all the time in this talk to convince you that where reciprocal connections are is meaningful. So, because if there are reciprocal connections there are simplices there, there's lots of stuff happening there, right? So the idea is we can use the density of reciprocal connections as a meaningful notion of sparsity. And why do we want to use this instead of simpler ones like simplex counts or edge counts? Because those are always correlated to the size of the neighborhood. And then it's very hard to take out the confounder of, is this tribe encoding more information just because it's larger or actually because it's more efficient? Reciprocal connection density seems to not be correlated with the size of the neighborhood. So it's a good density metric to test this idea. And we can test this by looking at it was mentioned yesterday, I think, the dimensionality of the activity space. So we can think of these neighborhoods as a readout, and we can look at the correlation matrix of this neighborhood, and then look at the principal component analysis. So I just have my correlation of activity of my neighborhood, and then I'll do the cumulative spectrum, like singular values, they're added, and I can ask what is the number of neurons that I need, or the number of dimensions that I need to explain 90% of the variance, and then look what is that dimension divided by the dimension of the tribe. So what is of the neighborhood, sorry. So what is the fraction that I need? And we will have something sometimes that look like this purple curve, where I need about 20% of the possible space to encode 90% of the variance, or we can have something like this blue curve where I need nearly 80% to encode 90% of the variance. So I think we can think of the purple neighborhood as being an efficient neighborhood. I don't need that much space to encode all of the information that is done in this experiment, while the blue one is not an efficient one because it uses a lot of the space to encode the same amount of information. And so we've done this, we've sampled many of these neighborhoods from the sparsest and the least sparse, where sparsity is this reciprocal density. And we find exactly that sparse tribes are highly efficient and dense tribes are highly inefficient. And we can test this in a different way, and I will use one more minute, which is let's use the work of some other people and let's see how we can use this to improve classifications via machine learning. So I will very briefly try to tell you what these people are doing in these two papers, but if you have more questions, three of the authors are here, so you can ask all the questions to them. So basically, you put stimulus into this circuit, and they say, how can we classify what stimulus has been given from the activity of the circuit? And I don't want to look at the activity of the entire circuit because it's huge, but just of a subset. So the way they do this is, again, they think of these neighborhoods as readouts. They look at the structure and they say, from the structure, I'm going to select some champions, the ones that have the highest, number of neurons, they have a lot of ways of selecting champions. So they select some champions. Then they featureize the activity of the champion. They again have many ways of featureizing that. And then they pass that to a simple SVM classifier to separate the stimuli. And when they do that, they get something like this. So here in the x-axis are the parameters used to select the champions. They have many. And here the different ones are different ways of featurizing one. I'm just showing you a sample. So here is a featurization based on the size of the activity. Here is a featurization based on topological structure of the activity, and a featurization based on spectral properties of the activity. What actually is happening in each of the featurizations, I think for this talk is not that important. The important part is that they have many selection parameters and many ways of featurizing the activity. And they have a lot of different accuracy values for these pairs. And what they conclude is for different networks and for different stimuli, there are different pairs that will work better than the other. But perhaps we can use now our knowledge of efficient networks and we say, okay, instead of just selecting champions, first let's select the sparse ones. We know sparse is efficient. Let's first select sparse and then let's continue their pipeline. We select the champions, we featurize, we classify. And when we do that, we see something like this. Suddenly accuracy is always better than just selecting champion because we are selecting the efficient champions somehow. And in gray here is if we would just sample that random. So like really understanding this kind of efficiency can also improve your machine learning pipelines to tell you how can you select things that are better suited because we understand what it means to be efficient. And I'm running out of time, but we're running lots of controls to see really further understand this. So I'm gonna stop there. So I hope I have given you an idea of how one can play with these and maybe use this to improve something in these concept-driven models. But I'll stop because I'm out of time. Thank you.
Go back to the last slide. I could not follow. Repeat, please. What is this like? What is it? A task that you network perform?
Okay, so no, so I should have said this earlier in the beginning. So these the blue-brain networks are not built to do a task. They're built to behave as what people observe in the lab, and then you see what happens. So what happens is you have this biophysically realistic network, and then you put a different kind of stimuli. Let's call them A, B, C, D, E, F. And you put them in different types. And then what they want is they want to create a classifier based on the activity of the network. So you put in the activity of the network and it tells you, oh, it's stimuli A that has been given. Oh, it's stimuli B.
What do? You train a linear readout of this or?
So that's what I was trying to explain. What they do is they will basically use an SVM classifier. And what I was explaining are what are the features that will go into the classifier. So the features that will go into the classifier is you take your different networks, your different neighborhoods, and you select the champion neighborhoods. So those that are structurally more important. And they have different selection parameters for those. So for example, the largest network, the network with the largest Petty coefficient, if you want some topology. The networks with the largest or the smallest directed connected components or whatnot. So you choose a subset of champions. And of these champions, now you have the activity on them, so you can featurize them. You basically, for each champion, I put really a black box here. For each champion, you have a vector of subgraphs of the activity, which you then featurize in a certain way, which are different ways of doing that. And then you pass that vector features to the SVM classifier. But the whole point is you have to select a set of champions, and you have to select which are given by a specific structural parameter. And you have to select the featurization, which is also given by a specific structural parameter. So you have a pair of this makes my neighborhood good, and this feature is a good feature. So you have to find this pair to pass this to the classifier. And what they find is which pair is better depends on the stimulus, depends on the network, depends on several things. And what I was trying to point out is if we do this champion selection by first sub-selecting the sparse ones, the efficient ones, then it doesn't matter which selection parameter you choose, and it doesn't matter which feature parameter you choose, you will always perform well whenever this feature is not zero. Yeah, this is a caveat. Does this make sense?
Yeah.
And these blue brain graphs are all the nodes the same, or do you have like inhibitor and excipitor?
No, there are many types. So there are inhibitories, there are excitatory types, but also within the EI types, there are different morphological types and there are different electrical types. So they have different parameters for the differential equations that are driving their behavior. So it's really a very complex model.
Right, and how did they how did this play into the role of the simplex?
It didn't. I think in the earlier paper, where they just look at 30,000 neurons instead of 4 million, they also did subgraphs of excitatory and inhibitory and also subgraphs per layer. And then you will see different characteristics of each layer, and also in E and I, you will also see a difference between them. We haven't done this for this huge one, mostly because it's huge. And it took really several weeks to compute just this one. Yeah.
I think in the interest of time, we'll save questions for the coffee break.
Yeah.
But let's thank Daniela again.